Hello students, I hope all of you are fine. I'm Dr. Manjoo and you are studying biotechnology with me. So today we are going to discuss the chapter cell and microbial cell culture. The continuation of this chapter, so we are going to discuss about the measurement of the microbial cell growth. So how to measure and the cell growth is take place. So first we are know what is the microbial growth. So microbial growth defined as an orderly increase in cell number and chemical composition of a cell, then we call it the microbial growth. Means if the cell number is increased and if the cell size is increased, then we call the microbial cell growth. So the growth is take place by the different manner such as the balanced growth. If the what is a balanced growth? Balanced growth means doubling of biomass is accompanied by doubling of their component. जितना उसका biomass बढ़ रहा है, according to that उसके component अगर cell के increase हो रहे हैं, then we call it balanced growth. So what is the doubling time? Doubling means time taken by any cell, for example bacteria, for increasing our population to double. Clear? So if the population increase is double, the time is called the doubling time. Yeah. So it's a time required by a cell to double its biomass or cell number, then we called it doubling time. So now now how we measure the growth of cells take place. For that measurement, what we do? First, we take the microbial cell culture. In the during the practical 11th class, we have studied how to culture the microbes in the lab. So first we prepare the media, then culture the microbes in the flask. So take the microbial culture in broth, and now, for example, if you're taking the bacteria such as the E. coli, so if you wants to see the how much amount of the E. coli cells are the produced by the culturing. So for that you can take the broth that containing the E. coli. So now centrifuge this cell and then obtain the pellet. So now drying the pellet and then weight the pellet. Yeah. So some precaution keep it in mind before the measuring the cell mass. First you have measured the blank weight of the. You can take the blank weight of the append of tube. Yeah. So it is measured through the research 1 billion cells equal to 150 milligrams. What does it mean? आपने आपका जो सेल मास है वो 150 मिलीग्राम आया है. It means that your pellet is containing 1 billion cells. So next method, it's a weight. You can see how much amount of the cell is growth by measuring the weight. And there is the next technique by which you can also find out the growth of the cell. It's the absorbance. So absorbance is taken through the spectrophotometer. I have already discussed with you how we take the absorbance in the lab, and as the absorbance is increased, that's so the cell growth is also take place. So here I have mentioned the growth of the growth curve of the bacterial cells, and so how to find out at a different time how the bacteria is growth. So it's the absorbance and it's a cell number. So as the absorbance is increased, the cell number is also increased. But at the stationary phase, there is no increasement of the cell number. So cell concentration of the unknown microbial sample can be calculated by measuring the absorbance at the same wavelength. Wavelength is remain the constant. So there is other method that is not explained much more in your syllabus. So we are just remember the name of the other method, such as weight of the cell measurement, turbidity measurement. We can measure the turbidity, So the spectrophotometer absorbance by the taking absorbance and the ATP measurements as the number of cells increase, the number of ATP is also increased. So next important method it's a viable plate count method. ना? So viable plate count means if you spread or grow the colony on a plate, nutrient agar plate, and then you can count how many number of colonies are comes in 1 microliter of sample. Yeah, you can first you prepare the plate, average plate. We have already done practicals for that, and after the solidification, you can take 200 or 100 microliter of the culture. It's depend on you. So keep it in mind if you taking the 100 microliter samples. So you mention the CFE colony forming unit in 100 microliter, and if you taking the 200 microliter, then you told it and the colony forming unit comes in the 200 microliter. So the Bible said how many colonies are produced on a plate. You can count it manually and you can calculate the CFE colony forming unit. There is another method. So next is the most important. It's also an instrument that is used for the counting the number of cells. So for it's a assembly of the counter method. So what happened in this method? So it's a nozzle of the machine, and you pass your cell suspension, bacterial suspension, and from this nozzle only single cell is passed. So once the cell is passed, it takes the or it generates the electrical impulse. If this electrical impulse is uh, recorded by the machine and find out how many cells are passed from this orifice. So you can find out the in which how many number of cells are present in the cell suspension. Taking 100 or 200 microliter cell suspension, how many cells are present in the 100 and 200 microliter cell suspension. But there is a drawback of this instrument. It is counting on both dead as well as live cells. Yeah, so Bible plate count method is most important. It is used for the Bible plate count. Bible means जो सेल जिंदा है उनका ही काउंट आप करेंगे इसमें Bible plate count है, but in case of the counter methods, so here this electrical impulse is generated by the both as well as dead as well as live cells. So it counts total cell count. Yeah. I hope everything is clear for you, and thank you for watching this video.